In which week we can see the development of brain vesicles? In which week we can see the development of brain vesicles? Vesicles by the 4th week. So how many types of vesicles we'll see? We have 2 types. What are those? What are the primary brain vesicles? What about the secondary brain vesicles? Okay, fine. What about the ventricles? How many ventricles are present? Four ventricles. What are those? Okay. 4th ventricle. These are the 4. We'll discuss about the foramens. Okay. So this is your telencephalon followed by diencephalon. Then we stop talking. Mesencephalon, then metencephalon. Last one is myelencephalon followed by your spinal cord. Okay. So what about the ventricles? This is your lateral ventricle. And what about the ventricle present here? This is the 3rd ventricle. Then in this region we have what structure we can see here? Aqueduct. Cerebral aqueduct. It is also known as your cerebral aqueduct is also known as aqueduct of Sylvius. SY aqueduct of Sylvius. Okay. Next. Which ventricle is present here? 4th ventricle followed by central canal. Okay. What fluid runs in this ventricle? It is the cerebrospinal fluid, where it will produce mainly. Your cerebrospinal fluid is mainly produced in say it louder. Subarachnoid space. Okay. Any other answers? Your CSF is mainly formed in lateral ventricle by the choroid plexus. Choroid plexus. Okay. So whatever the CSF produce here have to run in your ventricles present in the brain area followed by the spinal cord. Later it has to flow into your subarachnoid space also, and this flow or circulation of your CSF will happens through the foramens present in this area. Okay. So the foramen present between your lateral ventricle and 3rd ventricle, that is in this region, it is called as interventricular foramen. This is called as interventricular foramen. Okay, which is also known as Foramen of Monro. M-O-N-R-O, Monro. Okay, and your 3rd ventricle communicates with your 4th ventricle through your cerebral aqueduct. Okay, the remaining is your fourth ventricle and central canal. Your fourth ventricle is basically continuous with your central canal. Okay, so there will be no foramen, but between the junction of your hindbrain and spinal cord, we can see small dilation like this. Okay, this dilation is called as terminal ventricle. So these are the foramens we can see in the brain and spinal cord. Your lateral ventricle communicates with your third ventricle through the foramen of Monro. The third ventricle communicates with the fourth ventricle through the cerebral aqueduct. And your fourth ventricle is basically continuous with the central canal of the spinal cord. So between the junction of your hindbrain and spinal cord, there is a small dilation which helps in the flow of your or circulation of your CSF, and that dilation is called as terminal ventricle. Okay, so how this CSF will reach into your subarachnoid space? Through which foramen? Through which foramen your CSF will reach to the subarachnoid space? There are two other important foramen, that is Foramen of Luschka and Foramen of Magendie. Luschka is 2 in number, Magendie is 1 in number. These are the two foramens which helps your CSF to reach the subarachnoid space. Okay, these two foramens will be discussed in detail in your theory part. Okay, I mean the gross part will be taken on that time. For embryology purpose, you should remember intraventricular foramen, cerebral aqueduct and terminal ventricle. Okay, any doubts in this? This is all about the foramens present in your brain and spinal cord area. Any doubts? No doubts? This one. Foramen of Luschka, Luschka. It is 2 in number, and foramen of Magendie, Magendie. It is 1 in number. Any doubts? Any doubts in the foramen? This completes your development of ventricular system. All the ventricles along with the foramens detail you should be aware. Any doubts in this? Yes or no? Any doubts? No doubts. Okay. I'll give the notes. It will be there in PPT. Remember the anterior pore and posterior neuropore? Where have we studied the anterior neuropore and posterior neuropore? We have studied about it in the neurulation part. So in which week we can see the neurulation process? In which week we can see the neurulation process? What do you mean by neurulation? The process of formation of your neural tube is named as neurulation, and it can be seen in the 3rd week of the development. If you remember this diagram, what we have discussed, this is the yolk sac, amniotic cavity, and here we have the neural plate. This is the neural plate. Okay, and this is the intraembryonic mesoderm part. From this neural plate, your neural tube is going to develop. Okay, once the neural tube development takes place, this is how it is present in the initial stage. Okay, here we still have the amniotic cavity part. Okay, why it is still in contact or communicating with your amniotic cavity? What is the reason? These topics are already discussed in your previous class. Okay, by this week or in this stage, your blood vascular system are not fully developed. So your neural tube is dependent on the amniotic cavity for the required nutrients and nourishment. So it is still in the contact with your amniotic cavity. So this point is called as anterior neuropore, and this point, this is called as posterior neuropore. It is also mentioned as cervical neuropore and caudal neuropore also in few textbooks, but try to stick on to the word anterior and posterior neuropore. But by the end of third week, your neuropore or the neural tube will get separated from your amniotic cavity. Okay. So at which somoid stage it will get separate or it will get closed? Your anterior neuropore will get closed at the 20th somoid stage. Okay, and posterior neuropore will get closed by the 25th somoid stage. So the closure of your anterior neuropore can be seen by the middle of 4th week or 3rd week? By the middle of 4th week, and your posterior neuropore can be seen closed by the end of 4th week. Okay, this is the normal development. We have anterior pore and posterior neuropore. Stop talking there, last two tables. So in the 3rd week, it is still dependent on your amniotic cavity for the nutrient and nourishment purpose, but by the end of the 4th week, it will get separated from your amniotic cavity and present as a tubular structure. Okay, your anterior neuropore will get closed by the 20th somoid stage, that is almost by the middle of the 4th week, and your posterior neuropore will get closed by the 25th somoid stage by the end of the 4th week. Okay, but if this closure does not happen, it will lead to few clinical condition. Okay, if your anterior neuropore fails to close by the end of the 4th week, it will lead to a clinical condition called anencephaly. Okay, and if your posterior neuropore fails to close by the end of 4th week, it will lead to a condition called Rachischisis. It is also pronounced as Rachischisis. Okay, it is one of the severe form of your Spina Bifida. So what do you mean by Spina Bifida? Okay, spinal cord will get divided like into two sections. Okay, any other answers? Any other answers? What do you mean by Spina Bifida? One answer is your spinal cord will get separated into two structures or into two sections. Any other answers? Is it finished, this topic, Spina Bifida? Is it discussed in your gross part? Yes or no? Yes. No? Not yet? Anything about spinal cord it is discussed? What they have discussed about spinal cord? Just the gross part or what? Histology of spinal cord is finished. Okay, fine. Then just once check what is Spina Bifida. If possible, try to write a short note, okay? Because in most of the exams, Spina Bifida will be the short answers question. It is a very important question. Okay, so if your anterior neuropore close to fail, it will lead to anencephaly. If your posterior neuropore close to fail, it will lead to a condition called Rachischisis. It is one of the severe form of your Spina Bifida, or you can tell it is one of the form of your Spina Bifida. Any doubts in this? Any doubts? Okay. The two lateral ventricle communicates with the third ventricle via interventricular foramen, which is also called as Foramen of Monro. The third ventricle communicates with your fourth ventricle through the cerebral aqueduct. It is also known as aqueduct of Sylvius. The fourth ventricle is continuous below the central canal of the spinal cord, so it represents a small dilation at the inferior end. It is called as terminal ventricle. Okay. The cerebrospinal fluid is mainly formed in the lateral ventricle, and it leaves the ventricular system through the foramen of Magendie and foramen of Luschka into the subarachnoid space around the brain and spinal cord area. Okay, any doubts? These foramens are important for your MCQ questions. Okay. Clinical conditions: anencephaly, failure of the cephalic part, that is your anterior neuropore part of the neural tube to close, and associated defective development of the vault of the skull produces a congenital anomaly called anencephaly. Here the vault of skull will be absent. So which is the vault of your skull? Can you all point at the vault part of your skull? Just point it once, everyone. Where is skull? At least show me where is skull. Oh, okay. Where is vault of your skull then? Which part is called vault? This part. This is called as the vault of the skull. This whole area. From the top view, what we will see, no, this whole area is called the vault of the skull. So if a baby is suffering from anencephaly, the vault of the skull will be absent. As a result, your brain will be exposed to the surface area as a mass of the cell. Okay, brain is represented by a mass of degenerated tissue directly exposed to the surface area. Appearance of child is characteristic with prominent eyes bulging forward, and chin part is continuous with the chest due to the absence of the neck area. Okay. You can see here, this is how the baby suffering from the anencephaly will look. You can compare both the conditions. What difference you can notice here? Okay. Any other difference? Smaller brain, okay. This dotted line represents the normal skull area, okay. But here you can see the skull area present here. So the brain part, which is the degenerated tissue, is exposing to your surface area part. So this is how the baby suffering from the anencephaly will appear or look, okay. The next condition is Rachischisis. It is an incomplete closure of your caudal neuropore, that is the posterior neuropore, and defective development of associated vertebral arches cause a congenital anomaly called Rachischisis. Here it is usually localized in the lumbosacral region. Neural tissue is widely exposed to the surface area. Occasionally, the neural tissue shows considerable overgrowth. Usually, however, the excess tissue becomes necrotic shortly before or after the birth. What do you mean by necrosis or necrotic condition? Death of the tissues or cells is referred as necrosis. Okay. So this is how the baby suffering from your Rachischisis will appear. This mass is nothing but your neural tissue cells. Neural tissue cells. Okay. right after the birth, these tissues will get, the cell death will happen and it will be exposed to your surface area. By conducting a surgery, we can remove that surface, whatever the external part is there, no, this part we can remove, okay. So this is a baby suffering from the Rachischisis condition. Done? Okay. The next condition is hydrocephalus. What do you mean by hydrocephalus? Excess accumulation of fluid, okay. Enlargement of head area. Any other answers? Okay. So when there is excess accumulation of your cerebrospinal fluid in your ventricle area, it will lead to a condition called hydrocephalus. So when the hydrocephalus happens, your brain or your head area will get increased in the size. I can say it will bulge. But how the excess accumulation or production of CSF takes place? Because whatever the CSF will produce in your lateral ventricle will definitely flow from your lateral ventricle to central canal, and later it have to enter your subarachnoid space also. So how the overproduction of CSF is taking place? What is the reason behind it? How the overproduction of CSF is taking place? Obstruction. Absorption. Your CSF will get absorbed from other tissue. Okay, fine. Any other answers? Any other answers? There is in your gross anatomy textbook, there is a heading CSF circulation. Once go and see that CSF circulation part. Later I'll discuss how the overproduction of your CSF takes place. Formation. Yeah, from the yes, right. From the choroid plexus, the excess production of your CSF takes place. But how? What is the reason? It gets started overproduction and also obstruction in the flow of your CSF. Okay. So it is related to the choroid plexus part. Once go and see that part in your textbook. Later I'll discuss about that. Okay. So it is a clinical condition characterized by the dilation of the ventricles due to the excess accumulation of CSF within them. It occurs either due to the overproduction of CSF or by the obstruction to its circulation part. So you can see the picture and compare the structures. What is the difference you can see here? In the second picture, the ventricles are bulged or enlarged in the size due to the accumulation of your CSF. Okay. So this is how the baby suffering from the hydrocephalus will appear or look. So any doubts in this? Any doubts in foramens or clinical condition? Any doubts in the development of your brain vesicles, formation of fissures or development of ventricular system? No doubts? Okay. Then we'll go to the next topic, that is histogenesis. What do you mean by histogenesis? Histology of spinal cord is finished, right? So what are all the structures or what are all the cell structures you can see there? Whatever you remember, you can tell. Others? What cell structures you can see in the spinal cord? Have you observed the central canal while writing the histology? Have you guys seen the central canal in the histology of your spinal cord? Yes or no? So around the central canal you might have observed the white matter and grey matter also, right? So where we can see the white matter and grey matter? Outer part of the spinal cord. Which matter? Gray one or? Outer is white matter, inner is gray matter. Okay. Any other structure? So we can see central canal, gray matter, white matter. Any other structures? Any other structures? What about horns? Have you seen the anterior horn and posterior horn? What about columns? Anterior column, posterior column and lateral columns? Yes or no? If you have observed it in histology, say yes, otherwise fine. No. No? Okay. So this is the structure of your neural tube. Okay. So here we can see the fusion, and this fusion happens at the 4th somoid stage, right? So what is this part is called as? It is called as cranial part, and this is called as caudal part. Okay. So now we are going to take the section at the junction of your cranial part and caudal part here. Okay. So if I take a section and observe the structure, this is how it will appear. Stop talking. So now this space or cavity is called as dorsoventral cleft. Okay. And this part is called as roof plate, and this part is called as floor plate, and these two are referred as lateral walls. Lateral walls. Okay. So first we have taken the section between the junction of your cranial part and caudal part. If we observe the gross structure, we can see a cavity or an opening that is called as dorsoventral cleft. Apart from that, we have roof plate, floor plate and two lateral walls. If I see the histology of the same structure, this is how the structure will appear. Okay, so it is lined by which cells? It is lined by which cells? What cell structures you can see in spinal cord at least? Histology is finished, right? So this, whatever the cavity is present in the histology part, it is referred as neurocele. It is referred as neurocele, and this neurocele is lined by your pseudostratified columnar epithelial cells. Okay. In the gross part, this slit is referred as dorsoventral cleft. This is the gross structure. Okay. This is the histo part. Don't get confused. In the histology part, we are referring that cavity has the neurocele, and this neurocele is lined by your pseudostratified columnar epithelial cells. Okay. So now this pseudostratified columnar epithelial cells will undergo proliferation. Once the proliferation takes place, what types of cells we can see here? Since this is the part of your CNS, what types of cells we can see here? Your brain and spinal cord is the part of your central nervous system, right? The development is also takes place from the neural tube. So what type of cells we can see here after the proliferation? Nerve cells, very good. I'll write it as neuron cells. Okay. Neuron cells. Any other type of cells you can see? Oligodendroids. Okay. Axon cells or dendroid cells. Okay. Astrocells. Okay. Fine. Yeah, I said astrocells. The other one important cell is neuroglial cells, right? Neuron cells and neuroglial cells. These are named as neuroblast cells, and spongioblast cells. Okay. Your pseudostratified columnar epithelial cells will undergo proliferation and gives us two different forms or kinds of cells. Those are called as neuroblast cells and spongioblast cells. Okay. So whatever the structure present here is going to look like this now. Okay, this is the first layer. This cell layer is undergoing proliferation. So we can see neuroblast cells and also spongioblast cells. Okay, this is the spongioblast cells which is in irregular shape, and this is the neuroblast cells. That is not the cell structure. For our convenience study, I am writing it as a dot structure and irregular mass structures. Okay. So here it will form one more layer around the first layer. Okay. So this will be the first layer. Now we have got one more layer, that is the second layer. So whatever the neuroblast cells present here will undergo the maturation process. So how the maturation of your neuroblast cells will take place? General anatomy. How the maturation of your neuroblast cells will take place. This is the neuroblast cell. Okay. So during the maturation process, it will form a process on both the sides like this. Okay. Now it will go to the bipolar neuron stage. Okay. After that, one of the process will degenerate and only one process will remain. This is your unipolar neuron stage. Later, this unipolar polar neuron cell will give rise to the multipolar condition. Whatever this one process will remain, that is going to develop as the axon part, and the others, this is the dendroid. This is how the maturation of your neuroblast cells takes place. First we have the neuroblast cells. So on either side it will form a process and it will become a bipolar neuron. Later one of the process will undergo degeneration and we will be left with only one polar, that is the unipolar neuron stage. Later this whatever one process is remain is going to develop as your axon, that is the mature stage, or also we can call it as multipolar neuron stage. Okay. So now whatever the neuroblast cells present here will undergo maturation and it will give the axons like this. Okay. And few of the spongioblast cells will mature in this zone and few will come out of this zone and they will start get matured at this area. Okay. So now this will again form one more layer. So total we will get 3 cell layers. Okay. The very first layer, this is called as, what it is called as? It is called as matrix cell layer. Then the second one here, this is called as mantle cell layer, then followed by marginal cell layer. Is it clear for everyone? Any doubts? Any doubts? So first we have taken the section between the junction of your cranial and caudal part. If we see the gross structure, this is how the gross structure will appear. We have dorsoventral cleft, roof plate, floor plate, and on either side we can see the lateral walls. If I see the same structure in the gross histology part, we can see a cavity that is named as neurocele, and this neurocele is lined by your pseudostratified columnar epithelial cells. Later this pseudostratified cells will undergo proliferation to form two types of cells, that is neuroblast cells and spongioblast cells. Okay, so this neuroblast cells and spongioblast cells will together make the second layer, that is called as the mantle cell layer. So at last, whatever the neuroblast cells will get matured will again come out of the mantle cell layer along with the matured spongioblast cells, forming one more layer, that is your marginal cell layer. Okay, these are the three cell layers we can see in the histogenesis of your both brain and spinal cord part. Any doubts in this? No? Okay. The very first one is, what is the first layer? It is the matrix cell layer. The second one is mantle cell layer. Okay, the last one is marginal cell layer. Your matrix cell layer is formed by or lined by which cells? It is lined by pseudostratified columnar cells. This matrix cell layer is also referred as ventral zone. Ventral zone. Okay, your mantle cell layer is lined by which cells? It is lined by your neuroblast and neuroblast plus spongioblast cells. Okay, this cell layer is also referred as intermediate zone. Intermediate zone. Okay, the last one, that is marginal cell layer, it is lined by which cells? It is lined by axons of neuroblast cells plus matured spongioblast cells. Matured spongioblast cells. And this cell layer is also referred as marginal zone. Okay, so now imagine this has your central canal part. Okay, after the central canal, which area is present? Grey matter or white matter? After the central canal, which matter is? Grey matter. Okay, so this will form your grey matter. Okay, then it is followed by white matter. This is how the grey matter and white matter and also the central canal of your spinal cord and brain will form in the initial stage. Any doubts in this? Next we have histogenesis of the neural tube. The neural tube increases in thickness due to repeated mitosis of its epithelial lining. By the middle of the 5th week of embryonic development, the transverse section of recently closed neural tube reveals 3 distinct layers or zones. From within outwards, it is matrix zone, mantle zone and marginal zone. Okay, you can see the diagram. Just concentrate on this part. This will be your central canal part, or you can tell neurocele part. Okay, then it is formed by 3 different layers. Right after the neurosil, we have matrix followed by mantle, and the outermost one is matrix layer. Okay. So your mantle zone will give rise to or forms your grey matter part, and your marginal zone will forms the white matter part. Okay. And whatever the matrix zone is present, it will form along with your central canal. Any doubts in this topic? Any doubts in histogenesis? No doubts? Okay. Then we'll end the session here. Thank you.